Sugar versus brain health. Adults and young adults in the US consume, on average, 17 teaspoons of added sugar daily. This is over 70 grams of sugar, which is almost 2 to 3 times the recommended daily allowance. Altogether, this adds up to around 60 pounds of added sugar per year. Have you ever wondered how this may affect our health other than just our waistline? Today, I'm investigating the effects of sugar consumption on the brain and answering the question, does sugar intake contribute to a higher risk of dementia and Alzheimer's disease? As the most common cause of dementia, Alzheimer's disease is a major public health concern. Alzheimer's is a progressive neurodegenerative disorder that deteriorates memory, thinking, and behavior. Alzheimer's develops due to a combination of genetic, environmental, and lifestyle factors, but the exact cause is yet to be fully understood. Pay close attention to how Alzheimer's occurs in the brain. Alzheimer's occurs within the neurons in our brain. Inside the brain, there are proteins called amyloids. In Alzheimer's, these amyloids build up and clump together to form plaques outside the brain cells. These plaques block the cells from communicating with each other, making it harder for the brain to work. Inside brain cells, another protein called tau helps keep the cell's structure organized. In Alzheimer's, tau changes shape and starts sticking to itself, forming tangles. These tangles inhibit the transport of nutrients and signals, leading to the cells dying. As more brain cells die, the brain shrinks, especially in areas that control memory, thinking, and personality. This leads to memory loss, confusion, and trouble with daily tasks. Alzheimer's alone affects almost 7 million Americans, and as shown below, is projected to affect almost 14 million by 2050. While age and genetics are significant risk factors, researchers are investigating whether lifestyle choices like diet play a role, and sugar might be a key player. Sugar consumption has been linked to obesity, diabetes, and inflammation, factors that are also associated with brain health. But what does the data say about sugar specifically contributing to dementia? The following 3 studies will help us to gain an understanding of the current data on the effects of sugar on brain health. The first study, titled Associations of Sugar Intake, High Sugar Dietary Pattern, and the Risk of Dementia, is a prospective cohort study utilizing data from the UK Biobank, which encompasses 210,832 participants. This study analyzed over 200,000 participants with no prior dementia history, identifying high sugar dietary patterns using statistical techniques. 3 models were used to assess the risk of dementia across groups, refining results with each step. The study explored how sugar affects dementia risk based on age, sex, and genetic factors, and researchers looked at vascular disorders, metabolic factors, and inflammation as potential mediators between sugar and dementia. There are several important findings. The first being absolute sugar intake. Each additional gram of daily sugar was associated with a 0.3% increased risk of all-cause dementia and a 0.2% increased risk of Alzheimer's disease. The second finding, being about the relative sugar intake, was that a higher percentage of daily energy from sugar correlated with a 31.7% increased risk of dementia and a 24.9% increased risk of Alzheimer's disease. Finally, diets characterized by high sugar content were linked to a 9% increased risk of dementia. While keeping in mind that this was an observational study, so causation cannot be established, the findings suggest that excessive sugar consumption may elevate the risk of developing dementia, including Alzheimer's. This study is helpful in determining the role of dietary sugar in dementia risk and how high sugar diets affect overall cognitive health. By identifying these associations, this study can help us to create strategies for promoting better long-term cognitive health and prevent dementia through dietary modifications. The second study is titled Sugar in Beverage and Risk of Incident Dementia, Alzheimer's Disease, and Stroke, and is a prospective cohort study which uses data from the Framingham Heart Study, which is a community based in the United States. The participants' intake of sugar-sweetened beverages was assessed between 1991 and 1995. Then, the participants were monitored for the onset of dementia, Alzheimer's disease, and stroke, covering a follow-up period of 15 to 20 years. 63% of participants reported consuming no sugar-sweetened beverages, while 18% consumed 1 to 7 servings per week, and 29% consumed more than 7 servings per week. Using statistical modeling, this study calculated hazard ratios to assess the relationship between the exposure, sugar-sweetened beverage consumption, and the time to an event, dementia, Alzheimer's, or stroke. Risk associations were determined using the data. For a participant who consumes 7+ servings of sugar-sweetened beverages, the hazard ratio for dementia was 2.8, for Alzheimer's was 2.55, and for a stroke was 2.11. This study found a dose-response relationship, meaning higher consumption was associated with greater risk. Even at moderate consumption levels, there was an elevated risk of dementia, Alzheimer's disease, and stroke. Overall, this study demonstrates that the increased risk of these diseases applies across various levels of consumption, but the risk escalates with a higher intake. The final study is titled Intake of Sucrose-Sweetened Water Induces Insulin Resistance and Exacerbates Memory Deficits and Amyloidosis in a Transgenic Mouse Model of Alzheimer's Disease. This study is an experimental study conducted on a transgenic mouse model of Alzheimer's disease. In this study, male transgenic mice were divided into a control group and a sucrose group, receiving 10% sucrose-sweetened water. Both groups were fed a normal, low-fat rodent diet for 25 weeks. Throughout this time, measurements were taken involving metabolic markers, body weight and insulin levels, behavioral tests, Morris water maze and a T maze, and neuropathology, amyloid plaque burden and APO expression in the brain. There are several important findings from this study on the mice that were consuming 10% sucrose water. The first being the metabolic changes. The mice that were consuming sucrose gained 17% more body weight. They also exhibited impaired glucose intolerance and hyperinsulinemia, and plasma cholesterol levels were elevated by 30%. The mice that were consuming sucrose also displayed significantly worse performance when tested in a maze, showing impaired spatial learning and memory. And the sucrose consumption led to a 2.9-fold increase in amyloid plaque deposition and a 2-fold increase in insoluble amyloid in the brain. The ApoE levels, which is a protein promoting amyloid aggregation, were upregulated by 2.5-fold. This study overall highlights the potential link between dietary sugar and the exacerbation of neurodegenerative diseases. It suggests that chronic consumption of sugar-sweetened beverages contributes to metabolic disturbances and promotes the aggregation of amyloid beta, furthering cognitive decline and amyloidosis. The collective findings from these 3 studies reveal a compelling link between moderate to excessive dietary sugar consumption and the adverse effects on brain health. Given the evidence across human and animal studies, limiting sugar consumption should be considered a key strategy to promoting brain health and preventing neurodegenerative diseases.